Next talk is Jana Oliveras from the ICN2, and the title of his speech is Synthesis on Application Study of Iron Oxide Graphene Hybrid for Water Remedy.
Good morning. My name is Jana Oliveras. I am conducting my PhD in the Inorganic Nanoparticles group. I am willing to present today a synthesis and application study of magnetite hybrids altogether with graphene. And we are using this for an application in remediation. So let's start. Well, first of all, I want to talk about how did we decorate the graphene surface with magnetite. This is a method that we have been trying to develop during my PhD. So when we have particles in solution, we surfact them, as they have told before. No, we do this because of the synthesis. We want well-defined and stable nanoparticles. If not, we will not have them. So the issue here is that for creating the hybrid between the graphene and the particles, we need intimate contact between the both components. So these surfactants avoid the contact between them. So our approach here is to partially destabilize. Partially destabilize means that in a longish period of time, such a day or two, they would aggregate and it would be a complete mess. But if we work with them rapidly, they won't. For doing so, we have mildly destabilized them via centrifugation or centricons, depending on the particle size and properties. This allows us to find the mildest method we can think for each kind of nanoparticles. After this, we have incubated them with the carbon nanostructures that can be graphene or carbon nanotubes or graphene oxide. In this way, we have a slow depletion of the last traces of surfactant that we have. And afterwards, if we have an excess of particles, we will wash them, because if not, we will have aggregates of particles onto carbon nanostructures, and we don't really want that. By doing this, we can separate between the part that concerns the particle and the part that concerns the hybrid that we want to create. Thinking like that has allowed us to create a really wide array of nanocrystals onto different carbon nanostructures. We have synthesized nanocrystals in organic solvents, in aqueous media, we have synthesized semiconductors, novel methods, all of them with different shapes and compositions. And well, we succeeded and we have kind of universalized this, this approach. So when we first paired magnetite or magnetic particles with graphene and graphene oxide, we noticed that, as you can see in these videos, they were draggable with a magnet, such as Guba was talking us. And we thought, okay, we can, what we can do with this, because this is so interesting. So we can maybe absorb in the surface of the carbon nanostructures some kind of contaminant, and then we can drag them. We are geniuses, everyone does that. And so we thought about which kind of contaminant we wanted. So there is this kind of contaminant that are emerging pollutants. Emerging pollutants, you find them in really small concentrations in the nature. One case would be pharmaceutical drugs. So why we are concerned about that? Because yes, we use them, and then we go to the water treatment plants, and there is some residue that goes to our water, but also livestock uses them, and well, there is no as much control for this. And also, I want to talk about 2 points and open a little bit the discussion here. The first one would be that, yes, they are in really low amounts, and they really don't affect bigger organisms, but it can affect the whole trophic chain. When we learned about this, it was in National Geographic that there were some vultures that were poisoned by diclofenac. And vultures are not nice, we don't like them, but they can intercede in the trophic chain in a way that then we would have like other animals, carcasses all around. This would be very bad for the pollution of waters too. And also the second one is that pharmaceutical drugs are inherently designed to interact with living organisms. So if we have them in the water, what can we expect but for them to cause some damage to some species? So yeah, with that, what I mean is that yes, we have to remediate, but also maybe we have to design better drugs. And after saying this, let's go to experiments. So we chose ibuprofen and diclofenac. Ibuprofen and diclofenac have, as you can see, a very well-defined signal in the UVBs. In the case of diclofenac, we have a signal at 275 nanometers, and in the case of ibuprofen, it's a little bit more tricky because we have a signal of ibuprofen at 222 nanometers, but we also have a signal at 268 nanometers, which is from the temporary products. This we must remember for further explanation. And we firstly wanted to design the experiments. As you can see, first we developed some dose response experiments in order to know how the system would more or less behave. And then we wanted to optimize this hybrid even more via other surface variations that I will explain to you later. The experimental details are simply, we put the drugs and the hybrid together, we let them absorb, then we took the supernatant, and by that, we could measure which amount of ibuprofen or diclofenac we had in solution, and so value how much we had absorbed. It's quite easy, but it's also something that we thought that we could explore as a new method for being in field in the future. And yeah, in this first dose response experiments, we can see that the response was quite linear in both cases. And you can see in red, this is the EC50 concentration, which is the one that we were working with afterwards. There is differences, and a small difference between the graphene control, that is sol graphene, and the graphene magnetite hybrid. But this, then we tasted because we thought maybe it's because we have some because we have occupied space from the particles, but it's not like this. It's just that for the graphene hybrid, you have to work with the material and you have some losses in the process. So once we had characterized the behavior that the hybrid would have, we wanted to go one step further and put other variations in the surface. One of the variations was graphene oxide. We oxidized graphene in 3 days and 6 days under really acid situations. And what we saw is that in the case of diclofenac, we had a better response from the hybrid to absorption, but in the case of ibuprofen, it was worse. We think that it is because when you have graphene oxide, you add some electrostatic forces. So when you have only graphene, you interact via pi-pi stacking, but afterwards, the polarity of the compounds starts mattering. And diclofenac is more polar than ibuprofen, so its behavior was better in the case of graphene oxide. This was something that we learned. And then we wanted to go one step further. And we wanted to be able to reuse the hybrid. For doing so, we added titania to the equation. Because as Maria was telling us, titania is capable to react and with the photons and generating electrons. So we synthesized titania by a well-known method that we didn't develop, it's from someone else. And we can see here in the EDX that we had both titania and magnetite onto the graphene surface. So we had a multimodular approach to this. What we did was to expose again to the drugs, then we took this out and we added peroxide in order to have reacting oxygen species that would interact with the titanium. This would allow us to degrade the drugs that we had already captured, so that were still onto the surface of the graphene. And then we would expose it to the drug again, so we would assess if we were able to empty some spaces or not. What we've seen is that in the case of diclofenac, both in low amounts of peroxide or higher amounts of peroxide, the multimodal hybrid worked. And we can see that in this control that is only with the magnetite hybrid, it worked too, but only with the higher amount of peroxide. This is most probably because we are going through a Fenton-like reaction here. In the case of ibuprofen, we see that it didn't work when we had a lower amount of peroxide, but it did when it was higher. It's most probably because if you remember, we had these temporary products that are in equilibrium. So probably with the higher amount of peroxide, we got to break this equilibrium and displace the reaction. That's it. Conclusions, we have assessed the system and we have proven the synergy between the titanium and the magnetite and the graphene. And also the perspective, which is the most interesting part for me, is that we really think that it would be worse to recycle the titanium magnetite graphene hybrid, that is making several cycles of it and testing them in more realistic scenarios, because in the end we were working with buffer solutions. So thank you for your attention. That's it.
Thank you very much for this very interesting talk. Well, there are many questions.
Hi, thank you very much for a nice talk. I just have, probably it's a comment more than a question, because in the end you say that you were using environmentally relevant concentrations. They were milligrams per liter, correct?
Excuse me?
The concentrations of your pollutants were milligrams per liter, milligram per liter. Can you hear me well?
I am not hearing.
So, like this? The concentrations of your pollutants were milligrams per liter.
Yes.
And this is at least 3 orders of magnitude higher than environmentally relevant concentration. So this is just a suggestion for you. Maybe, I mean, I know, sometimes in the lab we are limited by the analytical methods and things like that. I think you should really try to work under more environmentally relevant conditions. These contaminants will be present in the highest loading wastewaters. This would be nanogram per liter,
yeah,
not getting even to microgram per liter. So I would say 4 to 5 orders of magnitude lower. And this is really important for your process because the mass transfer limitations look very differently when you go this low, So the amount of your catalyst that you need probably will be a lot bigger. But I think it's good to think about that early on, so that you're sure that you're really working in environmentally relevant context.
Yeah, this one is the EC50 concentration, but you are right that when in the studies, both ibuprofen and diclofenac are found to be in the nanogram liter. So yeah, this should be, of course, tested by HPLC probably, but yeah.
Thank you.
There's another question here.
Hi, thank you very much for your presentation. I just want to ask, you have a lot of absorption on the surface, and do you prevent it, or it's absorption that also degrades the diclofenac or ibuprofen? You have absorption on the surface, no? This absorption, how do you prevent it? Or it's like also the degradation with absorbing, it also degrades it?
We want for them to adsorb. So we are measuring them by the supernatant as if it was a yield. So it's more like the thing that we don't have in the supernatant, we assume we have in the graphene surface. And I don't know if I am answering your question.
Thank you very much.
Thank you.
Any other question? No? Thank you for your presentation.